Good afternoon. Thank you very much. So after this wonderful introduction by Michel, I would like to touch upon vascular neuroplasticity induced by exercise, and particularly on inter-individual variability of vascular or neurovascular plasticity. So as you have heard, we know that exercise can have effects on cognition, on brain volume, and it also has effects on perfusion. And this is particularly interesting because the animal studies indicate that neurogenesis in the hippocampus is tightly linked to changes in perfusion through angiogenesis. Putatively, this is because the same molecular regulators can basically induce neurogenesis and also angiogenesis. Now, in all the work that we have basically done and the work that's around there, as far as I know, none of these regulators such as BDNF, vascular endothelial growth factor, insulin-like growth factor have been shown to actually link to both perfusion and cognitive change. We had recently, this is also something that Michel had mentioned, together with Henriette van Praag, showed that cathepsin B is upregulated with exercise and related to cognitive change in humans. But again, this is not related to perfusion. So we are very interested in basically identifying something in the periphery that is linking both perfusion and cognition in humans. And we're also interested in understanding the inter-individual variability in perfusion changes after exercise. So in my talk, there will be two parts. First, I will talk about a protein called clotho, because this seems to be a protein that links actually both to perfusion changes and cognitive changes. These are data that are based on an analysis of serum samples from a study that we had already conducted earlier, and that's already published. And then in the second part, I will talk about inter-individual variability in vascular plasticity based on the vascularization pattern of the hippocampus. And I want to show you how vascularization patterns actually may affect the neurovascular effects of exercise, but also be related to cognitive performance in older adults. And this is a new study with an exercise intervention in young adults and also a cross-sectional study in individuals with small vessel disease. So the first part of the talk is on clotho, and this is based on this is based on a study that we have already published. This is an intervention study in old age where individuals aged from 61 to 76 years of age, healthy older individuals, exercised for 3 months, 3 times a week. And we had a control group that did muscle relaxation. And sorry, this thing is very stubborn. Do I have to point it to somewhere? Oh, no, okay, now it moves. Maybe I have to point it this way, okay. And we measured fitness level, as Michel mentioned, with spiroergometry. We obtained perfusion measures using gadolinium contrast-based perfusion imaging, and volumetric measures using 7 Tesla imaging, neuropsychological tests, which included verbal recall, but also spatial memory using the Ray figure. Interestingly, this is a recognition memory part that taps on into mnemonic discrimination, as mentioned by Michel. So in these published data, just to recapitulate, what we saw was that the exercise group, which are the black dots here, show a nice relationship between the fitness change and the change in the memory for the Ray figure and the recognition memory for the Ray figure. And the same holds when you look at changes in hippocampal perfusion and the Ray figure recognition memory and the hippocampal volume and this memory measure. And with structural equation modeling and path analysis, this suggests that hippocampal perfusion actually has a direct effect on recognition memory and also on hippocampal volume, and the volume actually does not explain additional variance in the cognitive change after exercise above and beyond the effects of perfusion. So this study indicated to us that perfusion has an important role in improving recognition memory. As I mentioned, we haven't found any relationship previously between BDNF, vascular endothelial growth factor, or IGF, and perfusion measures. And so we tried, so we were very interested in new developments regarding molecular regulators that may be related to perfusion changes. And Klotho is very interesting in that regard. Klotho is a gene that basically was originally identified in mice as a putative age-suppressing gene that extends lifespan when it's overexpressed. It induces complex phenotypes when it's disrupted that basically resemble human premature aging. When it's elevated in the serum of mice, it improves cognition, particularly hippocampal-dependent memory. And intriguingly, recent evidence suggests that Klotho is also a regulator of postnatal neurogenesis, affecting neural stem cell proliferation and sufficiently to actually also impact hippocampal-dependent spatial memory. So we thought maybe Klotho is related to increases in perfusion through its putative effects on neurogenesis. And we analyzed remaining samples from this study I just mentioned and looked at the plasma levels of Klotho. And it turns out that indeed the changes in the plasma levels of Klotho were nicely related to changes in hippocampal blood volume, so perfusion measures obtained with gadolinium contrast imaging. There was also a nice relationship with the change in the recognition memory score that I just mentioned. And when we again conduct structural equation modeling, it turns out that Klotho can be modeled to have basically a direct effect on hippocampal perfusion, and both jointly have an effect on cognition, and we don't, again, need the hippocampal volume to explain additional variance, or hippocampal volume doesn't explain any additional variance beyond these two factors on the cognitive change. So I think there is a complex story emerging that we still need to understand how these different neuromodulators affect cognition and perfusion, but this is the first time where we see a peripheral marker actually related to perfusion changes and cognition. Now, in the same study, and now I'm coming to the second part, we also, and I think this is something that we observe in most exercise studies, we were quite intrigued by the fact that in the exercises, some people actually show a decrease in perfusion and in hippocampal volume. And we also see that in studies in young individuals. And so this inter-individual variability, where some actually show an increase in perfusion and some show a decrease in perfusion, basically drove us to look into this very, much more closely over the past few years. And one way we followed this up was by looking at hippocampal vascularization patterns. So to that end, we conducted a study in healthy young adults to establish the methodology, basically. And here we have a similar setup as in the previous study with 4 months of controlled exercise, 3 times a week. But now we have ASL as a perfusion measure, and we have obtained a very high resolution 7 Tesla angiographic to look at the vascularization pattern of the hippocampus. We had an early measure after 4 weeks, and then a second measure after 4 months to look at the dynamics of the change as well. So just to show you that we get the expected fitness effects, you can see that the VO2 changes over the training, in the training group, but not in the control group. Serum levels of lactate drop as expected with exercise. And we also get a very similar effect on the Ray figure. So I'm going quickly over the slides because I want to come to the more important aspect, which is the vascularization. So we obtained time-of-flight angiographic images at 0.3 millimeter resolution using a slab centered along the long axis of the hippocampus. And the idea was that we identify, using the time-of-flight, the different vessels that actually supply the hippocampus with blood. This is what we see with the time-of-flight images. This is what you would see in a post-mortem study. And you can see that you can actually identify some of the major vessels that supply the hippocampus in the time-of-flight imaging. Now, hippocampal vascularization patterns have been of interest in clinical research, but also in neuroscience for quite a while. Early in this century, Uchimura discovered or reported that the posterior cerebral artery is one of the major sources of hippocampal blood supply. And you probably all know about transient global amnesia, where basically a temporary disruption of the supply leads to a global loss of episodic memory that is transient. Now, in addition to the posterior cerebral artery, there is also supply from the internal carotid artery, which is mediated through the anterior choroidal artery. It turns out that there is quite a variability across individuals in how much the posterior cerebral and anterior choroidal artery actually contribute. This has been described in quite detail in a study by Erdem et al. In the 90s. And it turns out that with the 7 Tesla angiographic data, we can actually replicate the exact patterns that he described in his study. Here you see basically the various patterns. There are 5 patterns that can be histologically defined. These are the population frequencies, and here in dark you see the frequencies that we see with 7 Tesla, which actually match this very well. The main message here is that you can divide the population into roughly 2 parts. So one part has a single supply from just the posterior cerebral artery, and the other individuals have a mixed supply coming from both the posterior cerebral artery and the anterior cerebral artery. And the hypothesis that we had was that if you have 2 supply sources, you may actually benefit more from the exercise and be in old age less vulnerable to effects of, for instance, vessel disease. So we first looked at in a follow-up study in older adults whether this actually has any clinical meaningful significance. And we did this in individuals with small vessel disease. So this is a study by my colleague Stefanie Schreiber and Valentina Perosa. She looked at individuals with cerebral amyloid angiopathy, mixed cerebral amyloid angiopathy and hypertensive angiopathy, or just hypertensive angiopathy, so 20 individuals in total, and compared them to healthy older controls, and characterized, again using 7 Tesla time-of-flight imaging, the hippocampal supply patterns. And she just classified individuals into those who have no hippocampus with a mixed supply, 1 hippocampus with a mixed supply, or 2 hippocampi with a mixed supply. And it turns out that when you look at cognition, so these are CVLT, this is a verbal memory test score, you can clearly see that if you have no hippocampus with a mixed supply, you are cognitively performing lower in this vulnerable group of individuals with with small vessel disease. So there seems to be a clinical significance to this. And now when we look at the ASL perfusion changes in the young adults, we see a an effect of the supply pattern on hippocampal perfusion. What's plotted here on the x-axis is the change in perfusion after 4 weeks, and here you see the change in perfusion after 4 months. And so anything above 1 means there's an increase in perfusion. And so here these blue dots basically mean that these individuals have an increase in perfusion after both 4 weeks and 4 months. And the green dots are individuals who have who don't have a mixed supply but a single artery supply. This is now plotted here for the right hippocampus. And you can see that you are much more likely to have actually no effect of exercise or even a decrease in perfusion when you have just, when your hippocampus relies just on one source of supply. So this is something that basically we are quite, excited about in terms of understanding some additional source of variability of perfusion effects of hippocampal exercise. So to summarize, I think Clotho is interesting because it shows a link between exercise-related perfusion changes and cognitive changes in old age. And some of the variability that we observe by way of the effect of exercise on perfusion, surely not all of it, but some of the variability may also have to do with the hippocampal supply patterns, vascularization patterns. And I would like to thank all the people who have participated, particularly Andreas Becker, who is a sports scientist, very gifted sports scientist, who has basically conducted most of these studies. Thank you very much.